So I said we're going to start using the word ICF and ECF, or extracellular fluid and intracellular fluid. Oftentimes, conversationally, we just say water in and out of the cell, but I need you to understand that we don't truly mean pure water. We just mean, the watery environment inside and outside of the cell, all the dissolved stuff. The ICF and ECF can have different solutes. They do have different solutes, different dissolved ions. That means they have different charges, positive and negative. And so because of that, that plays a large role in cell activity and cell conductivity. There's a little bit of an electrical charge to your cells. I can't remember if it's this chapter or the next. And so I'm just going to go ahead and talk about it right now. If we learn it again, who cares? We are going to learn that inside of the cell, we have to keep potassium in the cell. Potassium constantly wants to flow out. It's leaky. It flows out of what we call leaky ion channels, but we want to keep potassium. So the mnemonic device that I learned was Circle K, like the Circle K Ranch, if you're familiar with that Western story. But Circle K, potassium has to stay inside of the cell, but it's always flooding out. And so we've got to pump it in constantly. We've got to keep potassium inside the cell. Sodium, we always have to pump out of the cell. And sodium is constantly flooding in because that's how your brain sends signals down the cell line. But potassium, we got to keep in the cell. So don't forget that. When we're talking about the phospholipid barrier, because it's not just phospholipids, it's also cholesterol, glycoproteins, sugars, proteins, fats. They call it a fluid mosaic because it's made up of a lot of stuff, and a squishy sheet. It's a mosaic of molecules that's kind of fluid. You could think of it as a sea of lipids in which proteins float like icebergs. I think the textbook says that. And the proteins are like gatekeepers or bridges, but really it's just this, this area here, phospholipids, with a bunch of different proteins in it, either connecting cells to each other, acting as the fingerprint for your body to recognize, hey, this cell belongs to us, or acting as channels for things to flow in and out of the cell. We call the lipid bilayer we call the phospholipid membrane, the plasma membrane, a bilayer because it's actually two layers of phospholipids. The phosphate heads point to water, so we say they're hydrophilic. Remember that? Hydrophilic, hydrophilia. The tails, the nonpolar tails, are fatty acids, so they're nonpolar and they point away from water. They're hydrophobic. And so this creates this barrier. It allows us to have a definition between what's inside and outside of the cell. It allows for cells to have ordered structure. And so the phospholipid barrier is made up of lipids and glycolipids, which are just sugars and lipids stuck together. And what's important about these glycolipids is they may act as antigen or receptors. So when we say antigens, what we mean is a place that's going to cause an antibody to form. It actually means antibody generator. So each of your cells, everybody that is listening, everybody, everyone pretty much has their own sugar fingerprint, their own unique glycoprotein layer. That's how your cells know that you are you. Even if you have identical twins, the glycoprotein complex layer is going to be different. It's how your cells recognize you as you. If you're immunocompromised or if you have a disease like lupus, your cells may forget what your glycoproteins are and recognize you as a foreign invader. Or if a bacteria or some other pathogen is introduced that has a different glycoprotein, glycolipid layer, it says, hey, this shouldn't be here, and attacks it. Or if you have an organ transplant, you might have to be put on immunosuppressants, because even though you can match the blood type, the sugar fingerprint, the glycocalyx outside of that cell is always going to be different than yours. And so your body is constantly going to recognize it as, hey, that's not you. That's why it's so cool to think about, what we might come up with in the next 10, 20 years as we get better at growing organs. I don't know if you've heard that, we can grow pretty much your own, almost your own heart and things these days. And that's a lot better than transplants. One, because you won't have to wait for a transplant. And two, it will actually be made up of your own cells. So it kind of sounds like science fiction, but that's the next thing. I mean, once we can figure out how to effectively grow these things, we won't have to worry about different negative effects of, hey, your body recognizing that's not you and attacking it. Which that's kind of cool. And it all just comes down to these special sugars here. Notice these little ringed lipids right here. Those are cholesterol molecules, steroids. They give structure to that phospholipid barrier. So it's squishy, but you don't want it to collapse on itself. And so it stops these tails from sticking together. And so every so often you'll have some cholesterol. That's why you do have good forms of cholesterol. It's important in structure. Now the phospholipid layer is impermeable to water-soluble molecules. Fat-soluble molecules can fit through, but water-soluble molecules have to make their way through a protein. They cannot diffuse through on their own. So in this phospholipid layer here, we don't just have sugars, we also have proteins in two types. Integral, those that integrate and go all the way through, and peripheral, those that hang out just on the periphery, on the outside. Peripheral proteins often either stick two cells together or aid in cell conversation, and integral proteins act as channels or pathways for things to get inside of the cell. The integral proteins are part of the lipid barrier, and they can carry large molecules that couldn't fit inside or outside of the cell on their own. And so some things aren't going to form into, or aren't going to be able to, so I'm trying to do 2 or 3 things here at one time, aren't going to fit through that layer. They can't diffuse through, and so we have to have these proteins that act as gateways and channels. Pores is another form. The book sometimes calls integral proteins IMPs, or integral membrane proteins. I don't use that word a lot. I often just say integral proteins, but you'll see IMPs shortened a lot, and that just means the proteins that usually act as a channel, the ones that go all the way through. And so a cell has to be able to control what can move in and out of the cell itself, and these proteins act as that control mechanism. When we need things pumped in, we can activate certain pumps. When we need things pumped out, like sodium, we activate that pump. Sometimes it's the same pump that we just change shape. Some IMPs have carbohydrates attached to them, and so we call them glycoproteins. And some are receptors that react only to specific chemicals, like insulin. If you send out a hormone like insulin, you don't want every single cell in the body to absorb it. You only want it to be absorbed by the cells that you're trying to talk to. And so some cells have special receptors just waiting to receive that hormone. They can only fit that hormone. So let's just imagine, let's say that, I don't actually know, but let's say, insulin looked like that. On the outside of a cell, there could be a particular receptor that only that insulin hormone could fit in. And so once it hits that, then it can bring it inside the cell. But other hormones that are shaped differently couldn't fit in there. And so the cell's not going to accidentally grab onto something that it is not necessarily needing. Now, receptors are weird. We learn more about them as the semester goes on. But something that's difficult to remember and that has a major role in hormones, either with, let me take a step back. So your cells constantly are either making new receptors or destroying receptors, depending on if they need them. And they're extremely sensitive. Sometimes those control mechanisms break, or sometimes we wear them out. And so if we artificially put certain hormones in our body, so if someone is on steroids and they're taking testosterone, that's a huge amount of testosterone. Your body makes a ton of extra receptors. And then when you're not on testosterone, suddenly all those receptors aren't being used and they collapse, and you try to recompensate and you make a ton of estrogen. And it's just, your body tries to maintain homeostasis even when it doesn't, or even when it is kind of out of whack. And so just remember that we are making these cell receptors when we need them, breaking them down when we don't. And we will expand upon that story as the semester goes on.